Assalamu alaikum guys. Okay, I hope you're doing well. This is Dr. Shah Fahad Khan on your screen. Okay guys, so today we will be reviewing homeostasis as a chapter. ठीक है कि इसमें क्या क्या चीज़ें बहुत इंपॉर्टेंट हैं और वो स्टूडेंट्स जो MD-CAT और AKU के लिए प्रिपेयर कर रहे हैं एज एन अकेडमी उन्हें किन किन चीज़ों पर फोकस करना चाहिए. ठीक है, अगर वो अकेडमी जा रहे हैं तो ठीक है, वेल एंड गुड. अगर नहीं जा रहे हैं तो भी कोई मसला नहीं है. I'll इंशाल्लाह हेल्प यू आउट. ठीक है, सो बेसिकली होम्योस्टेसिस की डेफिनेशन आपको पता होनी चाहिए. ये होम्योस्टेसिस होता क्या है. ठीक है, फर्स्ट ऑफ ऑल होम रेफर्स टू a place where you live, right? Your home. Stasis refers to static, यानी कि stationary. So homeostasis refers to maintaining a constant home or a stationary home, or the definition would be maintaining a constant internal environment of the body. ठीक है, होम्योस्टेसिस. इसमें हमारे पास तीन सेक्शंस आते हैं जो कि हमें मेनली जिन पे फोकस करना होगा. उनमें नंबर 1 ऑस्मोरेगुलेशन, नंबर 2 एक्सक्रीशन एंड नंबर 3 थर्मोरेगुलेशन. ये तीनों के तीनों आपको आने चाहिए. ठीक है, ऑस्मोरेगुलेशन क्या चीज़ है और इसमें क्या क्या पता होना चाहिए आपको. ऑस्मोरेगुलेशन की डेफिनेशन, एंड अगेन गाइस बेसिक रूल ऑफ थंब है कि आप चाहे जो भी चैप्टर कर रहे हो, स्पेशली इन बायोलॉजी, आपने उसकी बुक रीडिंग लाजमी करनी है क्योंकि बुक्स की लाइंस के बीच से भी कोई भी क्वेश्चन आ सकता है. इट्स दैट्स हाउ इट वर्क्स. ओके, सो ऑस्मोरेगुलेशन में यू नीड टू रिमेंबर व्हाट हाइपोटोनिक, हाइपरटोनिक एंड आइसोटोनिक सॉल्यूशंस आर विथ रिस्पेक्ट टू अ पर्टिकुलर बॉडी सॉल्यूशन और विथ रिस्पेक्ट टू अ पर्टिकुलर सेल. ठीक है, अगर आप एक सेल को उठाएं, उस सेल को रखें और हमेशा याद रखिएगा एक इम्पॉर्टेंट बात कि ये टर्म्स जो हैं हाइपोटोनिक, आइसोटोनिक, हाइपरटोनिक, ये तीनों इन रिलेशन टू अनदर सॉल्यूशन होती है. ठीक है, जैसे अगर आपके पास एक सेल है और उसे आपने एक बॉडी सॉल्यूशन में रखा तो द कॉन्सेंट्रेशन, द सॉल्यूट कॉन्सेंट्रेशन ऑफ दैट सेल विथ रिस्पेक्ट टू द सॉल्यूशन इन विच इट इज़ केप्ट. हमेशा एक सॉल्यूशन को दूसरे सॉल्यूशन से कंपेयर करते हुए आप हाइपो, आइसो और हाइपरटोनिक का टर्म यूज़ करेंगे. ओके, अब व्हाट इज़ ऑस्मोरेगुलेशन. ऑस्मोरेगुलेशन बहुत सारे लोगों को ये लगता है कि इट्स द मेंटेनेंस ऑफ वाटर पोटेंशियल. ठीक है, वाटर पोटेंशियल मेंटेन होता है इनडायरेक्टली, लेकिन इस टर्म का मतलब ऑस्मोरेगुलेशन मीन्स it's maintaining the osmolarity. Osmolarity refers to solute concentration, not water concentration. Okay, it refers to solute concentration in a particular solution. तो आप एक solute के concentration को maintain करते हैं, water potential खुद ब खुद maintain हो जाता है. ठीक है, अब हाइपो, हाइपर और आइसोटोनिक की बात कर लेते हैं. आइसो का मतलब होता है सेम, तो हाइपो तो इनसाइड सॉल्यूशन जो सेल का होगा और जो जिस सॉल्यूशन में प्लेस होगा उनका सॉल्यूट कॉन्सेंट्रेशन इक्वल होगा, उसमें नो नेट फ्लो ऑफ वाटर इनसाइड और आउटसाइड द सेल होगा. ठीक है, सो नो चेंजेस इन द साइज ऑफ द सेल. ठीक है, हाइपरटोनिक कहेंगे हम उसे जिसमें सॉल्यूट ज़्यादा होगा. तो अगर आप किसी सेल को हाइपरटोनिक सॉल्यूशन में रख देते हैं तो बाहर सॉल्यूट ज़्यादा है, सेल के अंदर कम है, तो अंदर से बाहर की तरफ पानी जाएगा, एक्सो ऑस्मोसिस होगी. ठीक है, और सेल श्रिंक कर जाएगा. ठीक है, उसे अगर हम एक एनिमल सेल को रखेंगे उसे हम कहेंगे क्रीनेशन. अगर प्लांट सेल को कहेंगे तो प्लाज्मोलिसिस उस प्रोसेस का नाम होता है, जिसमें सेल मेम्ब्रेन सेल वॉल को छोड़ के सिकुड़ जाती है. फिर आते हैं हाइपरटोनिक के बाद आइसोटोनिक, हाइपोटोनिक सॉल्यूशन. हाइपोटोनिक मीन्स लेस सॉल्यूट कॉन्सेंट्रेशन, सो दैट मीन्स हाई वाटर पोटेंशियल. सो इफ यू कीप अ सेल इन दैट सॉल्यूशन, वाटर इज गोना एंटर द सेल. ठीक है, एंड सेल इज गोना increase in size, can swell up. If it's a animal cell, it's gonna burst. If it's a plant cell, it's gonna become turgid. ठीक है, फिर नेक्स्ट हम ये सॉल्यूशंस की टाइप्स आपको पता होने चाहिए. फिर ऑस्मोरेगुलेशन के बारे में बात करेंगे हम. ऑस्मोरेगुलेशन प्लांट्स में होती है. प्लांट्स की 3 टाइप्स हैं आपके पास, हाइड्रोफाइट्स, मीज़ोफाइट्स, ज़ीरोफाइट्स. तीनों के अडेप्टेशंस आपको पता होने चाहिए. ठीक है, टेक्स्ट बुक में लिखी हुई है, ज़्यादा डिटेल में नहीं जाऊँगा. फिर ऑस्मोरेगुलेशन एनिमल्स की बात करेंगे. एनिमल्स की 2 टाइप्स होती हैं. एक होते हैं ऑस्मो कन्फर्मर्स, जो कि अपना इंटरनल एनवायरनमेंट रेगुलेट नहीं करते. उनकी जो जिस एनवायरनमेंट में आप उनके सेल को रख दें वो बिल्कुल वही वाली कॉन्सेंट्रेशन उनमें आ जाती है, यानी कि फ्रीली सॉल्यूशन में एंटर भी कर सकते हैं और फ्रीली सॉल्यूशन से लीव भी कर सकते हैं. वो ऑस्मो कन्फर्मर्स होता है. और दूसरी कैटेगरी होती है ऑस्मो रेगुलेटर्स की, जो कि स्ट्रिक्टली मॉनिटर करते हैं कि सॉल्यूट की कॉन्सेंट्रेशन इनसाइड द सेल क्या है. ठीक है, जिसमें हमारे सेल्स, ह्यूमन सेल्स भी आता है. ठीक है, एनिमल सेल्स भी, कॉम्प्लेक्स एनिमल सेल्स भी आता है. ठीक है, तो जितने भी कॉम्प्लेक्स एनिमल्स हैं, हायर वर्टिब्रेट्स वगैरह, उन सब के ऑस्मो रेगुलेटर्स मोस्टली होते हैं, ठीक है। तो वो अपने इंटरनल एनवायरनमेंट को, सॉलिड कॉन्सेंट्रेशन को मेंटेन करते हैं और डिफरेंट होता है कंपेयर टू द सॉल्यूशन इन विच दे आर केप्ट, ठीक है। तो अगर इंटरनल एनवायरनमेंट की सॉलिड कॉन्सेंट्रेशन डिफरेंट होगी तो ऑस्मो रेगुलेटर कहलाएगा, और अगर सेम होगी तो ऑस्मो कन्फर्मर कहलाएगा। फिर मरीन और फ्रेश वाटर फिश और टेरेस्ट्रियल एनिमल्स की जो रेगुलेशन है, ऑस्मो रेगुलेशन, वो सब आपको करनी है, ठीक है। जिसमें फ़्रेश और मरीन वाटर के कम डिफरेंसेस आपको पता होने चाहिए कि फ़्रेश वाटर, क्योंकि फ़्रेश वाटर में होती है तो वो लार्ज वॉल्यूम ऑफ यूरिन प्रोड्यूस करती है, डाइल्यूट यूरिन प्रोड्यूस करती है, और इसके अलावा पानी उसमें खुद ब खुद गिल्स के थ्रू चला जाता है, लेकिन सॉल्ट नहीं होता तो वो एक्टिवली सॉल्ट को अपटेक करती है थ्रू द गिल्स, और पानी बिल्कुल भी पीती नहीं है फ्रेश वाटर फ़िश। लेकिन जो सी वाटर यानी मरीन वाटर फ़िश होती है वो पानी भी लार्ज क्वांटिटीज़ में पीती है, वो स्कैंटी यूरिन भी प्रोड्यूस करती है, एंड कॉन्सेंट्रेटेड सॉल्ट्स को एक्टिवली एक्सक्रीट आउट भी करती है, ठीक है। और पैसिव लॉस ऑफ वाटर भी उससे कंटिन्यूसली हो रहा होता है, तो उसे मेंटेन करने के लिए वो कंटिन्यूसली ड्रिंक्स वाटर, ठीक है। और कुछ एनिमल्स, फ्रेश वाटर के एनिमल्स ऐसे होते हैं जिनमें कॉन्ट्रैक्टाइल वैक्यूल्स होते हैं, जैसे अमीबा या पैरामीशियम हो गए। कॉन्ट्रैक्टाइल वैक्यूल्स का काम ये होता है कि वो एक्सेसिव वाटर को एक्टिवली पंप आउट कर देते हैं सेल से, वो सेल से बाहर निकाल देते हैं, ओके। Then coming to terrestrial animals. So terrestrial animals are very important. This is an important topic because and you need to know about the fact that terrestrial habitat is the most crucial and the most robust habitat because of the lack of water. So they have to conserve large quantities of water inside of them. They have to adapt to conserving water. Okay, you also need to know about the unsuccessful land animal, which is amphibian, because amphibians cannot prevent the water loss from their body, and they've got many. They don't actually have the adaptations required for staying on land, so they have to continuously go back to water again and again. All right. And terrestrial animals can tolerate dehydration because of the cuticle, because of the thick skin, keratinized skin. Keratin, because you know that it's insoluble in water, so it's going to prevent the water's diffusion from the or evaporation from the cell surface, from the surface of the animal itself. This ability to tolerate dehydration is called anhydrobiosis. You need to know that as well. Then coming to excretion in different in plants and in animals. Plants में आपको पैराग्राफ पूरा पढ़ना है। आपको ये भी पता होना चाहिए कि प्लांट्स में प्राइमरी ऑर्गन एक्सक्रीटोफोर्स यानी लीव्स होते हैं, जिनमें एक्सक्रीटरी मटेरियल डिपॉजिट होता रहता है, फिर ऑटम में वो लीव्स जड़ जाते हैं खुद ब खुद, ठीक है। एनिमल्स की एक्सक्रीशन का पता होना चाहिए। एक्सक्रीटरी प्रोडक्ट डिफरेंट किस किस एनिमल में होता है, क्यों होता है। अगर कोई एनिमल ऐसा है जो पानी में रह रहा हो तो उसमें एक्सक्रीटरी प्रोडक्ट अमोनिया होता है। वो इसलिए होता है क्योंकि अमोनिया बहुत ज़्यादा वाटर सॉल्युबल है, so it's gonna require a lot of water quantity to be excreted out in urine. But if there's an animal like insects or birds which live on land and they do not really have access to a lot of water, they produce uric acid as their major primary excretory product, because uric acid doesn't really require any water at all. Okay, it's the least soluble of the metabolic waste. Then coming to urea, which mammals, amphibians, sharks and bony fishes produce. It is less toxic than ammonia, but more toxic than uric acid, of course, because it's less soluble than ammonia, more soluble than uric acid, so it will definitely require some water to be excreted out of the body. And where are they coming from, each one of them? So everything over here is coming from amino group, and amino group is coming from two sources, either from proteins or from nucleic acids. You need to know about the urea cycle, how it is producing the two nitrogen containing urea molecules, CONH2 twice. Okay, you need to know about every single step of that urea cycle. Then in representative animals as well, just read it through all the way through cockroaches and make sure to know that. Then coming to excretion in human, the major part, you need to know about the entire urinary system. You need to know about the excretion, other excretory organ like the liver as well, and you need to know all the functions of the liver, like bile synthesis or heat production, or there's an entire table 15.1. Okay, you need to know everything about the functions of the liver, the synthesis of plasma protein, synthesis of glycogen, iron storage, LDL, HDL, etc. etc. Urinary system is also super important. The urinary system is composed of your kidneys, ureters, bladder, urethra. Need to know the basic anatomy. The kidneys are supplied by the renal artery, drained by the renal vein. Renal pelvis is connected to the ureter, through which the urine is drained, and then the renal artery and renal vein. Renal artery is coming out of the dorsal aorta. Renal vein is draining into the inferior vena cava. You need to know all of that stuff. Okay, and there's adrenal gland sitting on top of the upper pole of the kidney as well. You need to know all the anatomic relationships. Then apart from that, you need to know the internal structure, internal anatomy of the kidney as well. You need to know about the cortex, the medulla, all the pyramids, all the nephrons, the parts of nephron. But apart from that, after pyramids comes minor calyx, major calyx, the renal pelvis, then ureter. Need to know all of that. And you also need to know the fact that the nephrons, the structural and the functional unit of the kidney, is located mainly in the pyramids of the kidney, which are located where? They're located in the medulla. Okay, so from the apex of the medulla, urine is formed, from where it falls drop by drop into the renal pelvis, and from there it is drained out of the body. Okay, you need to know about the outermost layer of the kidney as well, which is the renal capsule. Outside of that is the renal fascia and the mesothelium. You need to know all of that stuff. Apart from that, you also need to know about the functionality of nephron and the number of nephron. So as long as the nephrons are concerned, you are a millionaire. You've got 1 million nephron per kidney, so 2 millions in total. Apart from that, nephron, the structure of nephron is important. The Bowman's capsule, the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, and then finally the collecting duct as well. Then the afferent arterioles, glomerulus, efferent arterioles, peritubular capillaries, vasa recta. Apart from that, you also need to know about the Malpighian corpuscle, which is a combination of the Bowman's capsule as well as the glomerulus. Then this was the entire structure. Now you need to know about the functionality of urine formation, how nephron forms urine. So the very first step is ultrafiltration, through which you need to know about all the different types, all the different components which are ultrafiltrated, which pass through the glomerulus. Okay, these include glucose, 100% passing amino acids, then fatty acids, sodium, potassium, chlorides, bicarbonates, urea, water. Yeah, that's almost it. Small proteins. Okay, large proteins are not gonna be leaving the blood. If they do leave the blood and they do end up in the urine, then that's actually a disease. Usually occurs in a kidney injury, for example, in nephrotic syndrome. That's something above your level. You don't need to know that right now. Apart from that, you also need to know about ultrafiltration. Why it's called ultrafiltration? Because of the multiple layers through which the solutes have to pass in order to enter into the lumen of the Bowman's capsule. From there, selective reabsorption is going to then take place. But you also need to know about the stuff that will not be ultrafiltrated, which includes red blood cells, the blood cells in general, the WBCs in general. Normally it will not be passing into the urine, because unless and until there's a urinary tract infection or something. Apart from that, plasma proteins like albumin will also not be passing. Okay, if albumin is lost in urine, then that would actually decrease the oncotic pressure of the blood, because of which third spacing will occur, edema will develop, swelling of the body will occur in the dependent parts usually, or it can be in the legs, it can be in the shin, it can be in around the tibia, malleolus, periorbital edema, and yeah, that's pretty much it. Then you need to know about selective reabsorption at the proximal convoluted tubule and tubular secretion around it. Tubular secretion, let me just cover that first. H positive ions are going to be secreted out of the tubules, peritubular capillaries, into the tubules. Okay, that's active secretion, and that's one process by which H positive ions are going from the blood into the tubules, and that's the only process where the H positive, where something is going into the from the blood into the tubules. Okay, that's it. And because of this tubular secretion, the pH is maintained at 5.5 for the urine. The pH is acidic because of H positive ion secretion into the urine. Apart from that, selective reabsorption takes place at proximal convoluted tubule, and the most reabsorption takes place at proximal convoluted tubule. You need to know that. That's an exam question. Glucose will be 100% reabsorbed, amino acids will be 100% reabsorbed, fatty acids will be 100% reabsorbed, proteins will be 100% reabsorbed, sodium, potassium, and chloride are partially reabsorbed because the rest of them will be reabsorbed later. Water is partially reabsorbed as well because the rest of it will be reabsorbed later, and we actually want some water in the urine as well. So that's why we won't be reabsorbing all of that. Some partially urea is also reabsorbed, by the way. Then these substances, once they are reabsorbed, and yeah, glucose will partially be reabsorbed only under the situation when the person is suffering from diabetes mellitus, because in diabetes mellitus you don't have enough enough insulin, so blood glucose level is not kept under check. Because of that, the kidney's ability to reabsorb all the glucose which is coming, the excessive glucose, is exceeded. So the kidney is unable to conserve all the glucose which is being lost into the urine because of the high quantity of glucose, free glucose roaming around in the blood. Okay, and that shouldn't be happening. Okay, glucose shouldn't be high, it should be kept under control. Then moving on, yeah, that's proximal selective reabsorption. Is called selective because only selected substances are reabsorbed, and reabsorbed because this is the second time it's being absorbed, right? The first time was in the during the after digestion, it was absorbed in the ileum, and this is the second time. That's why it's called selective reabsorption. After this, the fluid, luminal fluid, is going to enter the descending loop of Henle, which is permeable to water. So water is going to be reabsorbed over there. The reason why water can be reabsorbed over there is because the medulla has a salty environment, and that's because of a mechanism called countercurrent multiplier. It's called countercurrent multiplier because the luminal fluid is flowing downwards, but the peritubular capillaries, which is vasa recta especially, is taking the blood upwards. So first of all, it's countercurrent, so water will easily be reabsorbed back into the blood over there. Secondly, the ascending loop of Henle actually is actively pumping out salts into the tissue, into the surrounding medulla, keeping and the water that is being reabsorbed is immediately taken up by the vasa recta. So water is not allowed to stay there. This multiplies the gradient concentration in the medulla. Okay, this multiplies, because if water was there, then the concentration gradient would become less steeper, right? We need to keep the concentration, the difference of water potential steep, so that water is being reabsorbed instantly. So water is reabsorbed instantly into the vasa recta, and from the ascending loop of Henle, solutes are constantly being pumped out actively into the medulla to maintain that salty environment over there. So this reabsorbs more and more water, and then as it's ascending upwards, I already told you about the ascending loop of Henle. Ascending loop of Henle is impermeable to water, by the way. It only reabsorbs sodium, potassium, 2 chloride salts, and actively pumps them out actually. And then this water enters a distal convoluted tubule. Yeah, then after the ascending loop of Henle, distal convoluted tubule is the next part where mainly sodium reabsorption takes place, and that's the place on which the aldosterone hormone, which is a part of the renin-angiotensin aldosterone axis, acts. What is the job of aldosterone? Aldosterone is released to maintain the blood pressure of the body. If the blood pressure falls, aldosterone concentration is going to increase. Okay, so aldosterone is going to be secreted from the adrenal cortex, and then from there it acts on to the DCT, distal convoluted tubule, from where it causes more and more sodium reabsorption. And because of the sodium reabsorption, water also follows sodium, right? Water follows its solute, so it's going to be reabsorbed as well. And because of the increased hydrostatic pressure, the blood pressure will be returned back to normal. And then finally comes the collecting duct. After the distal convoluted tubule, it drains into the collecting duct. In the collecting duct, now mainly it's water reabsorption that is being taken place over there. Another hormone called antidiuretic hormone, which is synthesized in the hypothalamus, but it is stored temporarily in the posterior pituitary gland, is secreted when it is secreted when the blood water potential is falling. osmotic pressure is rising and we need more and more water in the body, then ADH is released. ADH acts onto the collecting duct. It causes aquaporin, which are water channels, to increase in the quantity in the walls of collecting duct. Because of that, more and more water is reabsorbed from the collecting duct into the surroundings, into the blood capillaries, and because of that, as more and more water is reabsorbed, less water is excreted out. Because of that, the urine becomes concentrated, and due to increased water reabsorption, water potential rises back to normal. Okay, that's the function of ADH, antidiuretic hormone, and it also is called vasopressin. ADH has a second name. It's also called vasopressin because it also can cause the constriction of blood vessels. And then finally, as after the collecting duct, urine starts dropping down via the apex of the pyramid into the minor calyx, from there into the major calyx, and then from major calyx into the renal pelvis, and from the pelvis finally into the ureter, then bladder where it's temporarily stored. Then as the moment you relax the external sphincter, boom, it goes out via the urethra. With this, the function of nephron and urine formation is completed. Then we've got problems and cures of kidney. So the problems would include kidney stones, and how are they treated? So kidney stones mainly occur because of calcium increase. Hypercalcemia is what we call it, right? Hypercalcemia means high blood calcium levels, and hypercalciuria means high calcium in urine, and hyperoxaluria means high oxalate in urine. Calcium oxalate are the most common type of salts, which are stones, which result in the salts that results in stone formation because they are not soluble enough in the urine, so they deposit as stones. Calcium oxalate are 70%, then followed by calcium phosphate, then finally uric acid stones. Uric acids are stones are radiolucent, by the way. They cannot be seen on x-rays. They're just 5%, by the way, and they are only made when you are severely dehydrated. Okay, and how are they treated? So basically, first of all, we can manage them conservatively by just asking the patient to drink more and more water. If the stone is small, it's going to pass out on its own. Otherwise, shockwave, extracorporeal shockwave lithotripsy can be done, in which ultrasound or x-rays are used to directly target the stone. It's broken down into tiny bits and pieces, and then it's why the urinary tract can be removed. If the stone is very large, then it has to be surgically removed. Okay. And then finally, coming to kidney failure. Kidney failure occurs when the kidney is unable to form urine anymore, and it's unable to get rid of the excretory products from the body, and that may result in chronic kidney. Maybe a cause might be chronic kidney disease, which may result in end-stage kidney failure. If that occurs, then the definitive treatment is kidney transplant. Okay, but until a person is trying to find a match, because the match needs to be important, and usually it's a close relative, a blood relative that can donate a kidney. Until the person is trying to find a suitable donor for the kidney, dialysis is a suitable option to keep the patient alive. Okay, but this is required if both the kidneys fail. If one kidney is function, person can live a normal life, no problem. Okay, dialysis of two types, hemodialysis and peritoneal dialysis. You need to know both of them. Hemodialysis can draw blood from the arm, take it into the dialyzer, purify it, throw it back into the vein, whereas peritoneal dialysis, inserting the dialyzer fluid into the peritoneal cavity via the catheter, and then allowing it to stay there for a while, and then draining it out via the same catheter. Okay, this needs to be done quite a number of times. With this, the chapter osmoregulation is completed. Now and excretion is also completed, by the way. We're going to now talk about thermoregulation quickly. Thermoregulation, again, hypothalamus is the thermostat of the body, is going to constantly be monitoring the core body temperature as well as the peripheral body temperature via the blood, basically. The blood that is coming to it. If it feels that if the body temperature is trying to fall, it's going to automatically it's going to start activate correcting mechanisms. Okay, what are the correcting mechanisms? If the body temperature is falling, it's going to activate shivering thermogenesis and non-shivering thermogenesis. In shivering thermogenesis, shivering occurs, musculoskeletal system starts contracting, the muscles contract, the person starts moving forward and backward, behavioral changes can occur, person needs to get more and more warm clothes. Apart from that, a person can metabolism increases, because metabolism increases heat production. Erector pili muscles erect, vasoconstriction of the blood vessels, arterioles in the skin occurs, because of which the heat is not lost to the environment. Okay, and the temperature classification of animals has to be done, by the way, before that, like poikilotherms and ectotherms and endotherms and heterotherms. You need to know all of that stuff. Okay, I'm not going into the details right now. We are talking about humans. Yeah, so this is what happens, and because of these changes, the body temperature is going to return back to normal. Okay, and if vice versa occurs, then again corrective mechanisms will activate, but those corrective mechanisms will also be the exact opposite, meaning the body temperature is going to fall. We are going to try to reduce the body temperature in that case, because if the temperature is rising, so sweating occurs, and metabolism decreases, vasodilation occurs, you start breathing heavily so as to remove the excessive heat via the water vapors of the lungs, etc. Okay, then what happens? So what's pyrexia? What happens in fever? Okay, by the way, this loop by which this temperature is brought back to normal is called negative feedback, and negative feedback is crucial for homeostasis and homeostatic mechanisms. Okay, homeostatic mechanisms can be controlled only by negative feedback, not via positive feedback. Negative feedback, by definition, is a response of the body to a change in the body against that change, basically to bring it back to normal. Okay, that's negative feedback. Okay, and what happens in fever, by the way, when the body wants the temperature to stay high, and that actually signals us that something is wrong in the body. By the way, another name for fever is pyrexia. Okay, you need to know this term. There are certain chemicals in the body which are secreted before that. If an inflammation occurs, or if a microorganism attacks, infection occurs, or anything happens, there are certain particles which are secreted by either that microorganism or by the body's own blood vessels, inkjet blood vessels, etc. They are called pyrogens. They're going to go directly to the hypothalamus, and they're going to increase the thermostat, meaning instead of keeping the thermostat at the normal body temperature, which is 36 to 38, it's going to increase it to maybe 39 or maybe 40. So because of that, the body temperature is going to increase, and the body will maintain that increased body temperature to signal us that something is wrong in the body and do something immediately. And fever is not a disease on its own. It's just a protective mechanism, actually, because certain bacteria can be killed with heating. Okay, when the body temperature increases, that their enzymes can die, but our own enzymes will be affected as well. So the underlying cause of fever has to be treated in that situation. Okay, so with this, the chapter homeostasis is completed. I hope this will help most of you prepare for your AKU as well as MDCAT. If you have any questions, just link them down in the comment section. And if you guys want any one-on-one tutoring or group sessions, you can join my AKU-MDCAT 2026 batch. Okay, this is Dr. Shah Fahad Khan. Hopefully see you in the next video, inshallah. Assalamu alaikum.